Right. Thank you so much. Thank you so much, Dr. Moses, for the anthem. So quickly, we'll be going into the technical session, and this will be shared by Dr. Toyin, my good friend. So we will be having basically 3 technical sessions today. And the first will be the presentation by Dr. Ifeanyi Ezonwumelu, followed by Dr. Jide Olubiyi. And also we'll be having the session we missed yesterday, Dr. Andres Giesel from IITA. And then we'll be having a presentation by our major sponsors, the MRFSS, Molecular Research Foundation for Students and Scientists. They will be making a presentation also this morning during the technical session. And that will now lead us into the abstract presentation. And finally, we'll end with the poster presentation. So I'll be quickly handing over to Dr. Toyin to take over the technical session and run the events for the day. Hello, Toyin.
Awesome. Thank you very much. Welcome everybody to yet another time to rub minds together as we share different research ideas. And I would like to quickly make an introduction for our first presenter for today, who is the person of Dr. Ifeanyi Ezonwumelu. Sorry, I might not be pronouncing the name very well, but I will try. Dr. Ifeanyi Ezonwumelu is an infinite, I'm not sure what this acronym is, Infinite Lacaxia Marie Curie, PhD fellow at Iris Cassier 8th Research Institute in Spain, and is focusing on development of novel HIV cure strategies. He holds an MSc degree in forensic science from the University of Lincoln in the UK, IUEM, Portugal, and the Universidad de Cordoba, Spain, and a BSc in Applied Biochemistry from Nnamdi Azikiwe University. Dr. Ezonwumelu has actively participated in research projects related to molecular epidemiology and phylogeography of hepatitis virus transmission in the Iberian Peninsula, and is now leading an international collaborative project on molecular epidemiology of multi-drug resistance tuberculosis in Nigeria. He has previously worked as a visiting professor at IUEM, Portugal, but continued working as a visiting researcher at the HIV Evolution, Epidemiology and Prevention Group, University of Lisbon in Portugal. He's affiliated with different institutes like the Virus Host, a research group, which is a virus host interaction team, and also the Iris Cassier Institute of AIDS Research, and the Hospital University Germans, Trias i Puyol Universidad Autónoma de Barcelona in Spain. Dr. Ifeanyi, I think that is easy for me to say. You have the floor.
Good morning, and thank you for the kind introduction. Just a minute to share my screen. Okay. So today I will be talking about forensic investigation of virus transmissions and phylogeography of infectious disease of public health concern. Phylogenetics refers to the study of evolutionary relationships between organisms of different or similar species using whole genomes or genetic fragments. In fact, Every living entity in this world is connected in the tree of life through a common ancestor. And pertaining to viral phylogenetics, we aim to determine the genetic diversity and the divergence of viruses, and also to determine their evolutionary history through time. So during viral epidemics, it is important to address several issues to guide appropriate public health response. But first, we need to identify the causative agents. Are there transmission drivers, intermediate hosts? And then define transmission chains and at-risk populations. And finally, assess if there are any transmission hotspots, taking into consideration important determinants of viral genomic epidemiology, such as evolutionary rates, sampling time and frequency, and available epidemiological data. So the first part, I'll focus on the forensic investigation of virus transmissions. Deliberate transmission of HIV is a criminal offense in several climes, actually. Usually in investigations of these kinds, the null hypothesis is establishment of no linkage between the viral sequences between the suspect and the victim, leading to exoneration of the suspect. However, if that is not the case, then we can proceed to infer the direction of transmission by examining the tree topology of the viral sequences between the suspect in red and the victim in blue. Tree topological relationships can be inferred in this way. We have a monophyletic topology indicating that both subjects were most likely infected by a common ancestor. A paraphyletic monophyletic topology indicating the direction of transmission from the suspect to the victim, and a paraphyletic polyphyletic topology indicating directness of transmission from the suspect to the victim without intermediaries. In addition, we can also infer the time of transmission if the viral sequences were ciliary sampled and possess sufficient temporal signal using Bayesian statistical analysis. To proceed with the phylogenetic reconstruction, we must first retrieve appropriate case controls with similar demographics as the case subjects, and also reference sequences from curated databases for subtyping. Then perform a multiple sequence alignment. Just a sec, Then perform multiple sequence alignment, subtype the sequences, perform a comprehensive quality control to avoid false positive relationships resulting from convergent evolution due to selective pressures from drug resistance mutation, recombination, hypermutation, etc. Only then can we proceed with the reconstruction of the phylogenetic trees using maximum likelihood and or Bayesian phylogenetic method. So the case that we'll be exploring today is a rather bizarre one because it bucks the trend when it comes to pediatric HIV transmission cases, which is usually horizontal transmission from mother to child and never father to child. So how did this happen? These news items were sequel to this work that we published in 2018. This is part of my master's thesis. So a quick look at the case history. In Lisbon, Portugal, a 4-year-old child was diagnosed of HIV-1 infection in late 2012, but born to HIV-1 seronegative mother. And the only suspect in this case was the child's father, who was diagnosed just a few weeks after the birth of the child in 2009. When we examined the case history, we discovered that the man usually bathed the child, and at that time was presenting with fluid-filled skin blisters, probably containing infectious HIV variants, making us to hypothesize that this might be the transmission route via contact with the healing umbilical cord of the child. To investigate this, we collected blood samples from both subjects at 2 different time points and sequenced HIV gene fragments, EMF, GAG, and POL for phylogenetic analysis. So here is the tree topology of the viral sequences for the GAG and the POL. The man's viral sequences are highlighted in red and that of the child in blue. And in green, we have Portuguese controls. Overall, we observe that both subjects formed a strongly supported transmission cluster of subtype G, but distinct from other sequences in the phylogenetic tree. Moreover, the paraphyly of the man's sequences relative to that of the child indicates the direction of transmission from father to child. And regarding the estimation of the time to the most recent common ancestor, our time scale of phylogeny indicates that both subjects were most likely infected almost at the same time, although the man probably was infected earlier than the child, as we see with these higher diversity happening in the man here. So at the end, no charges were brought forward, as this was highly unlikely to be a case of deliberate transmission. But we have to bear in mind that phylogenetic evidence at its best can only be used as a circumstantial evidence and never as a primary source of evidence in criminal proceedings. Moving on to phylogeography of infectious diseases. As before, we start with a multiple sequence alignment file, perform quality controls, check for temporal signal before we can proceed to Bayesian analysis. So what we can do in these aspects, Bayesian analysis is very useful to us because it provides us with reliable statistical estimates that can tell us how strong are the transitions from one location to the other. So in this case, we usually use a Bayes factor estimates. So because we can have transitions that are unidirectional, asymmetric, or bidirectional, which is symmetric. So it can be that you have the epidemic going from one source to the other and back and forth. So the case I'll be presenting today, I'll be presenting our findings from the most recent study that we did on epidemic history of hepatitis C virus in Spain. Here we focused on 3 important determinants of viral genomic phylogeography: transmission clusters definition, time-scaled phylogeny, and spatial distribution to enable us to characterize the epidemic history, origin, transmission dynamics, and the genetic diversity of HCV genotype 1A clades circulating in Spain. Here we observe that the paraphyly of clade 2. Could you try again? Oh, sorry, that was my. The paraphyly of clade 2 relative to clade 1 indicates that clade 2 is parental to clade 1 epidemic in this case. Moreover, when we build the time-scaled phylogeny, we saw that clade 2 was at least 4 decades older than clade 1, and clade 1 experienced an exponential epidemic growth between the 1980s and the noughties, coinciding with the harrowing crisis here in Spain. So here we can visualize the simulation of the spatial temporal dispersion of clade 2 epidemic in Spain, which most likely originated in País Vasco in the northern part of Spain and was endemic in this part for at least 3 decades before spreading to Galicia and other parts of Spain. And with the clade 1, recall that one of the earliest dispersions of clade 2 outside País Vasco was to Galicia. And here we see that with clade 1, this part of Spain became responsible for the onward transmission of these clade throughout Spain. However, when we made the estimates of the base factor for these transitions, we observed that certain inconsistencies with the simulations that I showed previously. As before, we had a strongly supported probability by base factor estimation that Basque Country was most likely the place where clade 2 originated in Spain, before moving down to Andalusia in the south of Spain and Madrid. And these 2 parts would later become responsible for the onward transmission of clade 1 throughout Spain, as we see with higher base factor estimates for these 2 locations in Spain. So yeah, it feels good to be back. Well, it started here in 2009 with introductory bioinformatics with the likes of Professor Ezeonu and co. And then later fell in love with phylogenetic analysis thanks to Nuno and Thomas, capped off with an exciting case in criminal investigation of HIV. And later pivoted to phylogeography of other infectious diseases. And now leading a project on defining the genetic diversity of multi-drug resistance tuberculosis in Nigeria with our patient enrollment centers in NTBLTC lab in Zaria and Naima, Lagos. Transcriptomics, it's another aspect of bioinformatics, but because of time constraints, this will be story for another day. This story wouldn't be complete without the support received from the members of my lab, Dr. Esther Bayana's lab in ESI Kaisha, the viral hepatitis phylogeography group covering Portugal and Spain, and the MDR-TB here in Nigeria, Portugal, Spain, and the UK. And thank you to the study participants. And thank you for your attention. I'll be happy to respond to any questions that you might have.
Thank you very much. I think we have a question from Dr. Samuel. Yeah, you can unmute and ask your question. Or probably the host can unmute Dr. Samuel.
I saw that Dr. Samuel raised his hand.
Yeah,
yes, okay. They just give me privilege to mute.
Oh,
thank you.
Okay, that's fine.
How are you? Thank you so much. Interesting. You know, I was just going through this stuff, software for phylogenetic beast. That's one that kind of discouraged me from phylogenetic. It was so difficult to use those days. But I think it's a lot easier now.
Yeah, it can be complicated, but it depends on the research question that you want to answer.
Yeah,
so if it is something simple, you can just use a beast 1. If it is more complicated, you can go for beast 2, but you have lots of applications that you can use within the beauty and the beast package.
You can say that again. Anyway, that's just the side note. So you mentioned something about using this technique to confirm direct infection from, or deliberate infection, as it were. So in cases of rape, where there's a rape, is there a cutoff time for you to do the test? I'm saying this in light of the recombination possibility and some of the things that might lead to false positives that you mentioned in the case. So that's my first question in terms of the time. The second question is that so phylogeny helps us to look at all the history, and one of the things I've used to argue for the SARS-CoV-2, and especially the rapid nature of the vaccine development, is to say that this virus has an history, so it's not new, as it were. But have we gotten to the stage where we can now have predictive, correct me if I'm wrong, phylogeny system, in which case we just have a variant in South Africa just 2-3 days ago.
Yeah.
Can I predict what is going to happen in the next couple of years? The virus has a short, not so big genome.
Yeah.
If I'm designing a vaccine, can I predict what's going to happen? Can I have that vaccine ready if there's a possibility of this mutation happen? Can phylogeny help me?
Okay, so I will start with the first question that you asked. In the case of rape, you wouldn't have like a time constraint, because with viral evolution, it is so it has like a bottleneck in such a way that the strain that infected someone, let's say on Saturday today, if you sequence that strain from the same host in a couple of days, it might look so different. But when you combine these viral sequences from one host to the other, you'll see that these will still be so similar to the one in the host. So even if it takes ages to do that, you can always find some link. But this is not like, it is not foolproof either, because that's why we said that you can't achieve the level of certainty that you have with a DNA fingerprinting, because for us to reach that reliability, you need to sample all infected individuals, which it's not realistic, it's not possible. So that's why in the criminal cases of these infections, although that you can demonstrate that the viruses are related, you also need other secondary pieces of evidence, like you have to look at the case history, the serological test and stuff like that. So it will help you to build a stronger case, but we can never use this as a primary source of evidence in a law court. And regarding the second question that you asked, yeah, let's say with SARS-CoV-2, actually what they are doing in the lab with the development of new vaccines is like they model, like they try to insert some mutations to the spike protein, which you just, you have like a model where you can do this in such a way that even if you have lots of mutations, when you build this tree, you can be able to see if there will be a lot of changes. But with SARS-CoV-2, it is not even evolving really fast compared to what we have with HIV. So we are so lucky that it is not as complicated as HIV. So that's the only good thing about SARS-CoV-2. Yeah, I know that we will be so concerned that it is changing a lot and it is highly infectious. But on the bright side, at least the vaccines that we have should work for some time being, I think. So.
Yeah, thank you very much. I just want to add one thing, is that it's very, very, very difficult. And I don't think it's possible to be able to predict a mutation. You can just, it's just because the human system is very complex. And so when the virus tried to evade the immune system, you can't predict how the virus is going to do it, right? In real time. So, and I think most what epidemiologists do is just to, if we're able to get enough data to get a system available such that once there's an outbreak, this data can easily be collected and sequenced immediately. Now, this is one way I think it's possible, but to predict if there's going to be an outbreak,
no,
it's extremely complicated. I don't know if I didn't see any further questions. Maybe anybody with a question?
Yeah, sorry,
all right.
Annie, thank you so much. And
okay.
I hope you understand when I call you Annie. Yeah, thank you so much. One, first, for agreeing to be part of this at the shortest notice I sent across to you. Thank you. And I was also very impressed by your presentation this morning. I wouldn't know. I'm a bit interested in our local isolates in Nigeria. So when I saw your hepatitis C, samples circulating in Basel, Galicia, and all of that. So I wanted to ask, do you have any plan for, providing our local data from where we came from? Basically Anambra itself, because the hepatitis there, we do not have any clear molecular epidemiology of these isolates. So I don't know what you think in this direction.
Thanks for the question, but it is more complicated than that. From my experience, it's difficult to set up like a collaboration, a network, research network, because one, you need to find people that are doing something similar to what you're doing, because if you're going to start this from scratch, it is a nightmare. At least that is my experience with TB, that we are trying to get the isolates from Lagos and Zaria. So you need to have like people that are right there in the field, because for me, I'm not there. So if I don't have access to someone that is there, there's someone that is willing to take charge of the local cluster, then it's going to be really difficult. So it's only if I see these isolates, that's the viral sequences online, maybe in PubMed, I can do something like this if it has not been done before. So,
Okay, I write you more on this.
Okay.
Yeah.
Thank you very much for your presentation.
All right.
So we would go to our next presenter for today. And I would quickly read out his profile. Next, we have Dr. Olatunji Olubi, who is going to be presenting for us right now. He holds a bachelor's degree in pharmacy from the Obafemi Awolowo University, Ile-Ife, and a master's degree of drug discovery from London School of Pharmacy in London. And he has a PhD in computer-aided drug design from RWTH, Aiken. He's interested in the use of computers for the identification of new lead molecules, as well as to unravel the mechanistic basis for the action of pharmacological active compounds. He also employs computational biophysics approach in investigating the conformation of micromolecular systems. Dr. Olatunji, you have the floor. Is it?
Jide, I think you could share your slide now.
I'm trying to figure out how to do that. Hello?
Yeah, we could hear you.
Okay. Good morning. I'm using my device, my mobile device to present. I'm trying to see how to do the sharing now. Can't find the button yet.
A bit complicated on mobile device. I think you have to look for a 3 dots if you can't find it.
Okay, I can see something share.
Yeah, exactly.
Okay.
Then at the background, the slides would have been open somewhere. Yeah, so that it could be an option for you to click. Yeah. Okay.
Okay. Thank you very much. Good morning, everyone. And I want to thank the organizers of this event for this. And it's very great to be here. And I thank Dr. Ifeanyi for that wonderful presentation. Just like the last question came, I was wondering we could have something like that in Nigeria too. You know, that's when we can have better benefits for some of these skills that we have developed over the years abroad.
Yeah.
Okay, my name is Molujide Olubeyi. I'm not a Latinji, and also my affiliation is no longer Forschungszentrum Jülich in Germany. I finished my postdoctoral project there about a month or two ago. So my affiliation is currently Pharmaceutical Chemistry Department, OAU. And I also have research link with Institute of Drug Research and Development at the Bogoro Center of Afeabalola University. They recently set up a very powerful computer graphics lab there, and it's exciting to work with them. So I did my Bachelor of Pharmacy at OAU in Ife before I went to do my MSc in Drug Discovery. I'm talking about this because I would like to talk about how I found myself in this path, although I'm more in structural bioinformatics aspects. I left the country then to do MSc because I wanted to become a synthetic chemist. I wanted to be in drug discovery, and I thought synthetic chemistry was the only way to go about it. But during my MSc in the UK, I was introduced to bioinformatics, computational approaches to designing drugs. At first, I was skeptical, but then I had the privilege of being taught by people working in the industry in actual, drug discovery research, and they were using computers. And so I fell in love. And after that, I would later go to Germany to do my PhD. And since then, I've been going back and forth between Germany and Nigeria. Okay, most of my work has been done at the Forschungszentrum Jülich, and they have some of the very powerful computational resources that you can find in Europe, and they are putting it to good use. One thing I've realized now is that the way we do research has changed. Even though some of us, we find it difficult to agree with that change. And this change had been ongoing for years now. I'm saying this, I'm talking about some of us from Nigeria. At first, when I came to the country, people were asking questions. Is it really possible to design drugs, to bring in new drugs, to evolve solutions using computers? Some people vehemently were opposed to it. And I've also been privileged to teach drug discovery to bachelor's and master's students, and many people that have been teaching these courses, you will agree with me that we always have this idea that discovering new drug takes a very long time, and it costs a fortune, costs a lot, sometimes more than 10, 15, 20 years sometimes. But this is going to be a little bit somehow. I was going to say, thank God for the COVID-19 pandemic, because a lot of things have changed since that happened. A lot of things have changed now within, like Dr. Samuel just pointed out, within few years, or I say few months rather, we started getting so much information about this new virus, the COVID-19 virus, what you would not have imagined, what you might not have been able to get even in 10 years. And this is mostly thanks to what informatics has helped to achieve. In the words of Levine and Rank, they said, a situation where you have a stretch of DNA, painting a picture of informatics heaven, you have a stretch of DNA that might just represent a novel drug target for treating a disease. And it can lead to, by using database queries, you can find information about the gene and its function, the tissue and circumstances in which it and any other variants are expressed, and importantly, the 3D structure of the protein or proteins encoded by it, then next, the structure of the ligands known to bind it, and most importantly, whether it is a novel discovery. If you look at how they have painted what an informatics heaven is, it actually covers every single area, every aspect of discovering a new drug. It permits me to come from the perspective of somebody trying to discover or to design a new drug molecule. And you will notice that everything they've said about informatics actually touches on every single aspect of what we have to do to discover new drug molecules. And in my group, we're trying to like make use of that in the development of new herbal medicine as well. Okay. So like I was saying about the COVID-19 pandemic, in December 2019, the WHO was officially informed about the development at Wuhan. And then just a few days after 7th of December, the virus was isolated. And then by 10th of that same month, we had preliminary genomic sequence solved. And by 24th, we had the first three-dimensional structure of the viral main protease solved by homology modeling. I'm bringing this up because for us in drug design, especially structure-based drug design, having the three-dimensional structure of the target, which is mostly protein, is like the must-have before you can even begin to embark on that project. I was really amazed when this happened that we didn't have to wait for X-ray crystallography or NMR or something like that. Bioinformatics was able to give us the first 3D structure. And before X-ray and NMR were able to catch up, we already knew so much about different proteins and different enzymes in the system in this particular virus. So the three-dimensional structure is what we must have before we can begin a structure-based design of new leads. And without that, we are just laboring in the dark. And I've put here what a 3D structure of a protein or of a molecule will look like for people that may not be working structural bioinformatics aspect. Is simply an information, a knowledge of what the coordinates, the XYZ coordinates of each atom in that particular system, what it looks like. We put figures to that. And by the time you have this for the entire protein, you know what it looks like, let me say in reality, in virtual reality, you can accurately search for new ligands that can bind it, you can modify it, you can carry out mutational studies to see how that affects the stability of that particular protein and every other thing that your research interest is focused on. But we have this gap that the rate of determining sequence of proteins is much, much higher than the rate of solving the three-dimensional structures. You do protein digest experiments and then you find the amino acid sequence that makes up the protein. We have that a lot. But it's been very difficult for people in drug design to utilize this information. And that's because we don't know how they fold, how this primary acid sequence, how it folds in reality. And until you do that, you will not be able to make use of it in designing new drugs. In fact, there are some quarters, they believe that the rate of solving this primary amino acid sequence is about 50 times greater than the rate of now determining a three-dimensional structure of this particular proteins. And the thing is that most of the methods have been used for a long time. Don't let me say till now. For a long time, I've relied on X-ray crystallography and solution NMR. Now we have solid-state NMR. And these are processes that take time. Sometimes it can take years even to get the proper crystal, particular protein before you can go ahead and solve it. And then sometimes by the time you artificially induce the crystallization of that particular protein, you may have affected what it looks like in reality. And a very good example is one of the projects I worked on during my PhD, which has to do with Alzheimer's disease. And we had to use the amyloid beta peptide. The structure available, they were actually not related to what we believe exists in vivo. And that's because this particular protein, it aggregates very fast. And to be able to make it to be in the monomeric form for them to be able to solve the NMR, to collect the NMR spectra for it, they had to artificially make that environment, the local environment of that protein, they had to make it to be as lipophilic as possible. So we believe that a 3D structure obtained that way doesn't represent what exists in human body. So we had to look for computational means of getting that. We're able to do that, and that particular project and what we have, let me say, discovered or studied from it is actually in, I think, phase 2 clinical trials now in Germany. Okay, so in the absence of these experimental methods, homology modeling, which is a structural bioinformatics technique, is what we use to reliably get a 3D structure of homologous proteins. And the only method of choice is to generate a reliable 3D model from its amino acid sequence. This method has really evolved so much, and we can reliably use them. And the word homologue refers to similarities attributed to descent from a common ancestor. And one of the reasons why HM homology modeling has been very useful is because evolutionary related proteins, they usually share 3D structures, even when you have differences in them, you look at differences in the amino acid composition. I mean, the 3D structure is more highly conserved than the primary sequence. And also small changes in the sequence many times don't really lead to very substantial change in the 3D structure. An example is the hemoglobin, the sickle hemoglobin, for instance. You have a single point mutation that changes, I think, glutamic acid to valine. That doesn't really change the overall fold of that particular protein. Okay. So there are different applications that we can have for homology modeling, like from my field, presently structure-based drug design. And sometimes we also use it to understand what is really going on in the particular protein system. One of the studies I would have liked to talk about, but because of some data protection, I'm not able to do that. We really have to use a lot of homology modeling to present the new structure of one of the core members of the brain extracellular matrix, and it's a work that we're about to finalize. I hope in the next few weeks it will be available. Okay, so homology modeling, you generate your models and then you want to use it for drug design. We may fall into that particular, let me say, error of not trying to check the quality. Yes, most of the software that are available, they give you scores of the quality, but personally and from experience, I always like to probe to make sure that it's actually structurally stable before we go on, so that we won't have spent so much time and so much computational resources, and only to find out that the model we have built is actually not correct. Okay, so there are different ways you can do this. You can use model quality scores. Like the simplest one is actually the RMSD. You simply compute the deviation of each atom, especially the CFR atoms of your predicted model from the template. And then you also carry out model refinement procedures, especially having to do with the templates. Most of the software now, like iTaser software, they use multiple sequence alignments. I mean, I think by default, iTaser uses about 10 different templates to be able to align your sequence. And then loop regions, they are quite problematic because they're very flexible. Yes, they're problematic structurally, but when it comes to functions, they're actually very important for a lot of the signal transduction process in the body. So we must find a way to be able to model them as correctly as possible. And then we also have the side chain refinements, because misplacing a particular side chain may alter your predictions. Okay, so there is a method that we commonly resort to. It's easy to use, but it's quite expensive, and it has to do with carrying out conformational sampling using molecular dynamic simulation of your particular model. Sometimes you build a model and you're very interested, you're very excited that the model looks good. And by the time you subject it to MD simulation, you find out that it falls apart, it's not stable. You check the RMSD or some other structural parameters over time, and you see that it is not stable. So you have to go back and probably look for new templates to use. Okay, MD simulation simply is it performs geometric optimization. In a way of speaking, is actually looking for the low energy conformations in your system. And I've put graphics of ethane here. I mean, you simply perform a rotation of the single bond joining the two carbon atoms. You can find how the energy goes up on the y-axis and how it comes down. MD simulation can help you to sample all these and more. But an MD simulation is not as simple as that. You don't just sample the torsional arrangement around a single bond. Like every single bond there, you want to check the bond extension and the compression. You want to check the non-bonding terms. Every aspect of this particular structure is actually captured in the force feed, which has the general form like this. Although the particular force feed your software is using may be different from this, but this is the general form that you have. So MD is very useful for performing confidential sampling of the model that you have generated from using homology modeling. And it's also very powerful tool that we can use by the time we are done with docking. I know a lot of us in Nigeria, especially, we perform docking, but most times what I've not always seen is people subjecting their docking predictions to molecular dynamic simulation or any kind of conformational sampling. A lot of times you can have
Sorry to interrupt you. I think you are running out of time. Maybe you can just try to round up in
okay. Actually, this is my last slide.
Awesome.
Okay.
Thank you.
So you perform MD simulation of the prediction, you find out that things fall apart. So we should find a way to go around the fact that MD is computationally expensive. And I know that we're in a resource limited country, especially in Nigeria. And I think one of the things we can do is to form functional collaborations with people abroad or even people in the country, and then to know how to make use of the resources to solve local problems. Thank you.
Thank you very much. Awesome presentation. Do we have any question? Yes, we have a question from two people. First, I think Nameka Nnamdi raised up his hand first.
I'm Dr. Samuel.
Oh, Dr. Samuel. Good to see you.
So I think, yes, in that order, can the host try to help us unmute them one after the other?
Okay.
Yes, thank you very much, Jide, for that presentation. Yeah, I just want to ask, in chemoinformatics, do you use, can you apply it to viruses? For example, in phage discovery, at a point we thought of finding a phage that we could, because most people now try to use phages and antibiotics, combination therapy. So we thought of, can we simulate the binding site and then the receptor site and then the ligand site, and try to, in the phage particularly, try to incorporate a drug molecule to it, such that when the phage will now serve as a delivery agent into the bacterium of interest. So I don't know if this, I've looked up in the internet literatures, I've not been able to find something like that. Is it something that is doable? So thank you.
Thank you. Is it possible to take all the questions? Or should okay.
You can take all the questions all at once. Okay, Dr. Samuel is next. Can we try to?
Okay, thank you very much. All right. Well, thank you, Dr. Jide. Nice to see you. I didn't see you. I'd love to see your face. Okay. Two questions I have. So we spoke about homology modeling. So let me speak to my daughter. Babe, can you please reduce the volume? Hello? Reduce the volume, please. Thank you. So you spoke about homology modeling, and of course, that's based on sequence templates. Sometimes you use multiple sequences to design that structure. Recently, there's been this AlphaFold that came into the picture, and I noticed that even Swiss, what is it called? Swiss target or Swiss model prediction. That's one that predicts
Swiss
structure.
model.
Okay, yeah, Swiss model. Now they are now indexing the structures from AlphaFold. So what's your take on AlphaFold in terms of the reliability of the structure compared to the usual homology modeling? That's the first one. Then the second one, we have limited resource for MD. And I mean, I've tried to do that. I've tried to look for platform, online web servers, a couple of web servers, but the time frame that will allow will be very short. And some people will not allow to publish because of that short sampling time. But now you also mentioned NMR structures, which are symbols of possible conformations of the protein. If a docking is done on an ensemble of structures with allowed flexibility, will that somehow give us, how will I put it now? It will not replace MD. Will it just give us a better picture of the docking? I don't know if you get what I'm saying.
Yes, I do.
To limit it, because people don't have resources to do that. So, but find a way around it. Thank you.
Okay, thank you, sir.
The last person, Ranu Emmanuel, I think. Can you try to unmute yourself?
Okay.
Yes.
Thank you, Dr. Jide, for that wonderful presentation.
Thank
I have
you.
a question. The question is, I was told by a researcher abroad that molecular docking is no longer a good tool for it to use, and as well that for you also that molecular dynamic simulations need to get up to 100 nanosecond simulations. I don't know how true that is. Well, because in Nigeria, we don't even have the capacity to run up to 10 nanoseconds, talk less of 100 nanoseconds. And then he's saying that after the molecular, instead of molecular docking or after the molecular docking, you need to do MMPBSA to be able to validate your docking values. So I don't know what you have to say about that.
Okay. Yeah. Thank you very much. I will start with Dr. Samuel's questions, whether it is possible to simulate a whole phage. In principle, yes. Although personally, I don't work with such systems, such are very huge systems. But in principle, anything can be simulated. The only problem is that some of the computer wares that we have right now cannot handle such huge systems. And that's why we've been trying to reduce everything to the smallest form that our computers can handle. For instance, this is not really phage, but the system I went to work on for the last 2 years in Germany now, by the time I was done building the systems, I used homology modeling with MD simulation of different fragments of that system. And by the time I was done and I surveyed the system, it had more than 300 million atoms. And honestly, I've not been able to find any system, any platform, anywhere that can handle that. And I don't think that is as big as simulating like a whole phage or a whole virus. So I think we get there, but we're not there yet. And that's why we have to keep being on the lookout for development in computer wares and to see how we can scale up what we have right now. Okay. And then Dr. Agiye's question about the AlphaFold. Yes, I got to know about the AlphaFold, I think recently. Was it this year or last year? I can't remember when it came out. And that's because I was working on the homology modeling of my system. This system is so big, I had to cut it into almost 17 different fragments. And each fragment, by the time I performed MD simulation, was more than 1 to 2 million atoms. Okay, so when my professor introduced me to AlphaFold, that we should see whether it is going to be useful. Yes, it's very useful, but I also think everybody should check how relevant and how useful it will be in your system. Like my system, for instance, they have models that are already constructed for the system I was working on, but I found out that they were not able to simulate the environment, the protein environment. So they stimulated everything as hydrophobic. So it collapsed on itself. You know, I had a system that had to be linearly arranged, but by the time they simulated the entire protein sequence for the very big system, everything was collapsed. The model they had was collapsed on each other. And we know from experimental data and information over the decades that that particular system is not like that, it's elongated. But because they could not put in all that is required to ensure to enforce the elongated structure, it was collapsed. So it's, in my case, it's not useful. But in every other case, I'm sure, especially for very small protein system of less than maybe 500 amino acid residues, it's going to be a very great resource. And it's something that I look forward to using personally. Okay, you also asked a question about MD resources. I mean, whether we can use NMR structures. I mean, there's some from NMR structures, and then use that to replace some of the things we could have gained if we had used MD. Yes, the answer to that is yes. I mean, one of the work that we are doing right now, and I had to rely on 20 confirmations from NMR. So there are two things that I generally use MD for when it comes to virtual screening or ligand docking. The first thing is I use it to diversify the starting structure of the protein to be able to have, reliable ensembles to dock against. I prefer to dock against 10, 20 or more conformations of my receptor than to use a single one. I mean, of course, I will also include the crystallographic structure or the homology model structure there, but I prefer to first perform MD simulation, then do cluster analysis or maybe PC analysis, identify the important conformations of that protein, and then dock my ligand against all of them. And then I will now report the average of the affinities. Okay, then after the docking also, I also use MD simulation to see whether the dock ligand is going to be stable, maybe for 100 nanoseconds, for 500 nanoseconds, as the case may be. So the second part, the post-docking screening, yes, we are missing out on it, but the fact that we're using MD ensembles for docking, I think it improves the quality, at least from the study we did last year on COVID-19 research, where we were able to identify correctly. We went into the lab, identified correctly some inhibitors of the main protease enzyme. This was the method we used. Although we did not use NMR, we used MD-generated ensembles at the beginning. Okay, and then the other question is from Dr. Emmanuel about whether molecular docking is no longer relevant or not. I don't really understand the context in which that person said those words, but it is not true. In fact, if you just start checking the publications from last year till now, many of the inhibitors have been identified against different proteins of the SARS-CoV-2. They started with MD simulation. We have experience not just doing the docking, but also proceeding to the lab and getting results. I mean, based on most of the results we've gotten, we have more than 50% prediction rates from our predictions, ligand docking predictions. But you said something about the requirements for MD that is long, like 100 nanoseconds. Yes, it is true. If you don't have MD that is like 100 nanoseconds or so, they may not accept your publications. But then it also depends on the protein system you are studying. If you're studying a very small protein system, a folded system, 50 nanoseconds can be enough. But if you are studying a flexible protein or something that is huge, they may even require you to go for 1 microsecond. I submitted the publication before, I think more than 10 years ago, and I did 1 microsecond MD simulation, and they required me to elongate it by more than half another microsecond, 1.5 microsecond. So it depends on the system you're talking about. But what I would just encourage is that we should really start building our own platforms in Nigeria, especially. And sometimes you just need about few computers, good computers, and then you serially connect them, and you can do 100 nanoseconds, you can do 200 nanoseconds. Thank you very much.
All right, thank you very much for all of those questions and the response. So next, we will go straight to what we have on the agenda, and it's a presentation on Molecular Research Foundation for Students and Scientists Fostering Molecular Research in Southeast Nigeria. I believe Dr. Moses would make this presentation, if I'm correct. I think so.
Jide might need to stop sharing.
Yeah, you need to stop sharing.
Okay, sorry about that. Okay.
Awesome.
Okay. I wouldn't know if the slide is clear. Could you see my slides? Can I go on?
Yeah, we can see your slide. Yes,
okay.
Please go on.
Okay, thank you once again. So I do this presentation on behalf of the MRFSS. Of course, I serve as the consultant scientist to this particular foundation. And so in 10 minutes or so, I should be done with this, So the Molecular Research Foundation currently resides in a Chisco building in Nnamdi Azikiwe University, Oka. And this is an outcome of passion of few scientists, myself inclusive. Then after my PhD bench work in 2016 in Germany, I came back to Nigeria, and it became obvious that it could be possible that in a few years, if we don't continue in what we did outside, somehow we might also start relearning again all the exposures that we had. So we felt that we have to push to create a platform where there could be a continuity in some molecular exposures that we had, both for ourselves and also for the coming generation, as it's challenging actually to get the current generations, convinced that through molecular researches, they could have a fulfilled career. So the Molecular Foundation Research for Scientists and Students, of course, started with first as non-profit organization. And the aim is to foster molecular research among students of all levels, scientists, and those who are particularly interested in the field of molecular biology. So that was in 2018, and by this group of visionary scientists, with a passion to replicate knowledge and improve ease of molecular research in Southeast Nigeria. Now, these are the guys, of course, that came together. Of course, most of us, we know Professor Charles Esimone, who is the current Vice Chancellor. Of course, by then, he is, I think, the Dean, and also in the course of the discussion, before he became the DVC academic when this was established. So the guy at the middle is Mr. Kevin Udoyo, who currently is a nurse in Washington, D.C. in US, and myself. So, and of course, he is the major funder of MRFSS, and it's a way to help the environment. And maybe if it's here, I'll particularly say thanks to him for making the MRFSS, something that is currently running, and many persons are benefiting from this. Yeah, we have an objective. Of course, one is to enable access. So at the point that MRFSS came, there was no functional molecular lab per se in the Southeast Nigeria, obviously. Okay, we rely most on IITA Ibadan, or maybe in University of Ibadan to run some molecular biology. I've traveled to Osun State to use a molecular lab in Ladoka Akintola University, and this was not fair. So, and we felt that it's nice to first start, not to rely on government to do this, because maybe by now we are still pushing one letter forward and back, but on a personal ground, and those who are enthusiastic to push to make molecular biology facility available in Southeast Nigeria. So that was number one aim. Then we also felt that there are some students around that this lab could support, and they strengthen their career interest in molecular biology, and to support them. Of course, our eyes were also on indigent students who have this intellectual capacity in this STEM area. Three also was to provide employment for young people in the university. So we have a scheme of when some of them come around in the lab and they work like 2 hours or so and get paid at the end of the month, and to support their stay in the campus. Then also for the academic staff who are in the area of molecular biology. So it's also a good platform for them to collaborate in a sense, and they have some research questions answered. Our host is Nnamdi Azikiwe University, so we have an MOU where they provide the enabling environment for us to thrive, and where we do all of this, to foster molecular research in the environment, I mean Southeast in general. Okay, so we have outreach programs. Number one, we have Master Scholarship, which runs for 2 years. And currently the girl who is Mrs. Judith Nwachukwu, I think, concluded her own, she was the first to have this scholarship, and this ended in November 5th of this month. That is why you could see some applications also, adverts going around. And of course, we have excellent applications so far. We'll talk about that at that point. So this runs for 2 years, and to engage a student who just finished BSc and want to humbly stay in the lab and build capacity, build capacity beyond what a workshop could also do. Then we also have pre-doctoral internship. Sir Benjamin is from Nasarawa State or so, and he finished his own pre-doctoral intern. And in 2022, we shall advertise again for this position. But currently, we are looking for the master's position, someone else that we take over from where Judith Nwachukwu stops. So we also have a postgraduate intern. This runs for 12 months, as the case may be. And all of this is to strengthen the molecular capacity of the young people, and to somehow help them to see a particular direction to move in the course of their career. So we also have IT students that stay with us 6 months, and they will ensure that at the end of the season, basically they should be able to first understand the principle of PCR and also to run PCR. I started running PCR as a PhD student, but I saw that from my exposure in Germany that this is wrong, where we have students, bachelor students coming to do classical experiments with us in the lab. So I think that was why we have to push this down to undergraduates and to have some of them stay in the lab, learn basic things around molecular biology, and see if at the end of the day, they could pursue this as their career. So we have these 3 guys. The last 2 guys are girls has finished, and currently Wasolo BD is on and going to finish in January. So we also have Ms. Tomiwa Oladere, the resident scientist. Of course, this is the one that teaches most of the people that come for one of these programs or the other in the lab. Then under the outreach program, we have what we call the annual free basic molecular technique workshop for final year students in Southeast Nigeria. This started since 2019, and in 2021, of course, you know that 2020 was Corona year, and this was highly interrupted, so we could not really, have this workshop. But we have, apart from 2020, it held in 2019 and 2021, hopefully with the support of Nkaba and some other persons. So this will continue to happen every year. So we also have a new idea on how to further step molecular biology down to the secondary school, those in high school. As I also monitor the German system, I'm here, but I'm also looking at their system and the way it works. Those who do coding start doing coding from high school. Those who do learn some things that we struggle at the current level where we are to do, they start quite on time to build this foundation. So I felt that to start pushing this by way of summer school for those in SS3, maybe staying with them for 2 weeks, and both theory, both practical, we have a way to strengthen their foundation for future challenges in molecular biology. And we are also doing our best to encourage all those in biosciences, zoology, botany, that even if you're reading zoology, there is much to do in zoology. You could key into bioinformatics, you could key into molecular biology, you could just go on. Today, the solution to the COVID-19 virus, pandemic we are having, is all stems from bioinformatics and molecular biology. So, and anyone could key in to this. Okay. So in order to continue to strengthen the capacity around the environment, we also have to accommodate the scientists and postgraduates, maybe first those who were teachers in molecular biology. Our experience have shown that many teachers do not understand the basis of this. And sometimes when they want to forcefully convince their students to do a research project in molecular biology, it's difficult. And when they don't understand this, they oppose what they should not oppose. They ask questions that reveal that they are quite ignorant in this. So we failed to extend this to the scientists and also to the postgraduates so that they could be properly guided as they design some basic research in molecular biology for their students. So this we started in 2018 June, and that was the inaugural workshop to kickstart the MRFSS within Nnamdi Azikiwe University. So September, same year, because of high level of interest and subscription, we also have to host another. And of course, we don't take much persons because everyone must be responsible in the course of the workshop, starting from your starting material up to your amplification and up to other things you ought to do with your own sample. So not really a group work, but you have to have your own sample and flow through onto downstream applications with the DNA you have already extracted. Yeah, so we went also in December, October 2018 to learn how to write with Orode, who is a postdoc in Oxford University, who came around and engaged us for full one day on how to write scientific papers. It was quite interesting, and we've dwelt so much for some years on the basic molecular, and we felt we have to start advancing. Of course, we had that in July this year when I came around. I think, it happened, and a lot happened in the course of these outreach programs. Okay, research output from MRFSS, because we have some collaboration with the current lab I am in Germany on sample collection, and also possible to do SNPs, single nucleotide polymorphisms, to track the chloroquine resistant gene and also genetic diversities, and also Cage13. Cage13 was the one we currently published still in malaria journal, why genetic diversity based on glutamate-rich protein was in American Journal of Tropical Medicine and Hygiene. So this was the one we currently published in this November. Of course, most of us should know about Cage13 and also the associated alleles that are responsible for artemisinin resistance. Yeah, we're also designing another work to have a clear clinical studies around this, where we are going to expose to some persons on ACT, not just on artemisinin, maybe the monotherapy, but also the combination therapy, and also track the resistance. Recently in Uganda, of course, some alleles that are witnessed in Cambodia and Miami has also been reported around there. And we hope, as my interest is very much in my own environment, Anambra particularly, so that we could have some data and to reflect that's where we are doing some work around. MRFSS have also engaged greatly in webinars, starting from optimizing your PCR. So you could see that we are still on the basics in the sense that I know our environment. I know that many people are still ignorant of what we're talking about. And I know we have to dwell such on the foundation so that guys can stand strong in the course of time. So many people perform PCR and it fails, and there are challenges around it. So my colleague in the current lab where I am, Miriam Brody, who is a PhD student who is doing a lot of PCR on Plasmodium malaria, I engaged her to teach on optimizing your PCR for basic malaria research. So at the end of the webinar, we're also to see that, of course, what she thought was like the general PCR optimization, which cut across anything that has DNA or DNA you want to optimize for anything. Okay, Dr. Taisikwe is a senior scientist in the Institute for Tropical Medicine where I am. Also very good in qPCR, and also have developed novel techniques for HRP2 and HRP3 deletions in Plasmodium falciparum. So I also engaged her, and we were able to have this webinar, and the video is still available. You can also write and request for the video of this training and see how to optimize your qPCR for basic, research. Okay. Of course, many of us would have seen this as I announced that the girl who is the Pioneer Masters student with us, she has finished, and to that we needed to publish again. We have already received more than 104 excellent application with 22 first-class persons that apply for this position, which have almost confused us on where and who exactly to pick for this position. But we hope we'll be able to narrow at the end of the day. Yeah, what do we want to achieve? What do we want to achieve? Someone that is consistent, someone that will focus on molecular biology and learn the dynamics of molecular biology at what level. Also, we collaborate with some, IITA and also South African and Kaba and so to sequence, for some basic bioinformatics analysis that we also employ. But we'll see how it goes the moment this guy or this girl resumes. I think there will be 2E that already will take this position because of the high number that is also that apply for this. I will also say at this point that we felt that there is still a big gap in Nigeria, and maybe government could do more, and the scale up these things that I see that there are people at the background, first class and some people who need some of this position because we cannot do more than we have the capacity currently to do. Okay, this is our future event, maybe in collaboration with the University of Tübingen, where I'm currently a postdoctoral fellow. We are going to have plasmid design. Maybe because in the course of my postdoc, I have to now go into expression of many plasmodium falciparum proteins, AMA1, CSP, and all those antigens that plasmodium falciparum express at some certain stages of their life cycle. And the guy here, Dr. Rolf Svendel, is a senior immunologist, and also he is a group leader of the immunology team. You may not know, maybe in the course of coronavirus, I have to join his team to express RDB proteins that we are using to run some local ELISA and on SARS-CoV and all of that. So this meeting will come up, not this year. We are going to stay shout about this no longer because we needed to sit down and to find appropriate time for him to be disposed to
to cut you, Dr. Moses. You ran out of time.
Finally, what we support. We call it support because, it's not really a profit, guided outfit, is to support. So from DNA extraction up to bioinformatics and up to ELISA, all of these are fully subsidized, and some are also free depending on the collaboration. If also you are researching on some area that some of the guys within MRFSS is interested, then maybe we can discuss on collaboration level and run the samples for publication. Thank you. I don't know whether I exceeded 10 minutes.
Thank you very much. We would have just one question, and I think I see the hands of Dr. Samuel. Yes.
Oh, thank you, Dumala. All right.
Yeah.
So, a couple of questions in one, if possible. So,
All right.
so the first thing is that you started this as a public-private partnership with the university, or is it just a consultancy arm of the university? I just want to clarify that. And you said it was a non-profit, but you are now moving to profit, in which case you are getting to standardize your lab so that you can run samples for people that want to use it, if I understand where you are going.
Yeah, we never moved from non-profit to profit. It's still non-profit.
Okay.
Yeah, it's still non-profit, and we under the university, there's what they call BOT, build, operate, and transfer. So within some couple of years, we are going to be directly responsible for this. Then after, when the agreed years is expired, Then the university and some team members, we have what we call 30%, 70% management.
Okay.
But now it's still 100% managed by MRFSS.
Oh, by the university. Okay.
No, by the MRFSS, not the university. In university,
not by the university, but the university has a share in it. Okay.
Yeah. So, but in the scholarship program, the program is with the university because we offer the scholarship while the course is taken in any of the biological sciences.
Okay. They take the cost center for that. All right. So the second question is about reagents and kits. How do you source them? Do you source them directly?
So the chairman of the organization resides in US, so he has registered with some of the foreign companies where we get some of those and send across to Nigeria. That is why we could run at the price of as the subsidizer. That is why it's easier for us to do DNA extraction as a crazy price, and just at the cost because we get it cheap from outside there. I also have support from Tübingen University in some of the reagents that we still use around there.
Oh, that's so cool. So that leads with my last question, which is looking at talking to companies, because I know when I was in Novartis, I was able to get some of the things from them. They did commission some equipment over time, and they are always willing to ship if you can cover the shipping cost. And I'm sure even in your lab there will be equipment that are decommissioned.
Yes.
So we can, because I'm thinking that also, I mean, if you can just put a paper together, this would be nice for what you're doing with this organization, and that would be a nice one to really push towards that and use to get some of this equipment, at least from these companies. Good work. Thank you so much.
Okay, thank you so much.
God bless all of you.
Thank you very much. And so we would move straight away to our abstract presentations. And right away, we would have Dr. Nnaemeka Emmanuel. He's a lecturer at the Department of Microbiology, Plateau State University, Bokos. His research interests focused on the use of computational biology, genetics, and model system to study the evolution, diversity, and pathologies of pathogens that affect humans in plants in Nigeria. His lab is also interested in understanding how yeast are adapting to climate change in the environment, as well as tracking the transmission pathway of antimicrobial resistance in the environment using the One Health approach, and to advance the use of phage therapy in managing resistant bacterial infection. He obtained his PhD from Nnamdi Azikiwe University, Awka. He's an ardent supporter of reproducibility in scientific research. And so, Dr. Nnaemeka, you have the floor.
Yes, thank you very much, MRCF, for the opportunity to present and for a wonderful conference webinar. Yes, I don't know if you can see my slides. Can you see my slides?
Yeah.
Hello, can you see my slides?
Yes.
Yes.
Okay. Yes. My name is Nnaemeka Emmanuel Nnadi. As Judy was talking about drug discovery and how fast chemoinformatics is, I've used a little bit of chemoinformatics also in COVID research. I think phage therapy in drug development pathway seem to be the fastest and the easiest. So I will be talking about Pseudomonas falciparum, naming the virus. You will see, I was actually named after my daughter Zikora. Okay, so why phages? It's important to understand that WHO had said that by 2050, there will be about 10 million deaths. And what makes this interesting is about 90% of these deaths will occur in Africa and Asia. And also, it's projected that by 2030, antimicrobial resistance could force up to 24 million people into extreme poverty. Now, what we also know is that drug development takes a long time. And with even all the processes of drug trials and all of that, it takes a long time for a drug to come into being. And one of the things we also know is that before a drug comes into being, most of the organisms have developed resistance to it. So it now raises a public health challenge for us that's now brings in question what phages are. So we live in a time in history where everybody thinks the viruses are bad, but we have good viruses, and phages are one of the good viruses that we have on ground. So we have and I know that phages can be an alternative to antibiotics because, again, one advantage it has is that it's
yeah, internet, yeah. I think he had internet issues, yeah. So what we would do, whether to go to the next speaker or we wait a little? Yeah, I don't know. Limiting factor. He's back.
Sorry about that. My internet had issues. So one of the things we know about phages is that it's very effective, and a lot of compassionate studies are going on now for treatment, the use of phages for clinical treatments. An advantage it has is that, unlike antibiotics, phages are highly specific. So phages, you could have phages against Pseudomonas, phages against Salmonella. There are very few phages that are broad spectrum. They are toxic, although, but it doesn't. Most studies have also found out that it does not affect the microbiome of the system, and they are natural and abundant. And another thing important about phage is that it is self-replicating. So once you just give a single good dose, it keeps working until the pathogen is off, and resistance is very low to anti-phage therapy. So over the years, you could see phage genomes that have been, from 1997, there has been an exponential increase in phage genome that have been deposited in phage banks. In isolation of phage, in Paul Hyman's paper, he did a fantastic job with regards to talking about how phages can be isolated. So if you want to know about phage isolation, you can read it up from Paul Hyman's study. Now, but generally, for Pseudomonas phagocytora, we got it from soil and sewages and from the hospital environment. We performed an enrichment method using Pseudomonas aeruginosa as indicator organism, and so you could see effective plaques here showing that, oh, these are viruses that have grown. So from these viruses, we extracted our DNA. So now that assuming that you have gotten your own phage and you want to name the phage, it's important that phage naming is very important, because you don't want to get something that when people are talking about it, it will be confusing and all of that. So the International Community of Taxonomy of Viruses have come up with a communication on how to name and classify your viruses. You could see that by Andreasen's paper. One of the things you need to do is that before you identify your phage, it's important to use phage name checker to check the name of your phage if somebody has used it before, and it's also important to use the complete genus name. So you could see Pseudomonas phage, then the name, a unique identifier attached to it. Now, another thing you have to understand when you want to get your phage is that the question, does my phage represent a new species? There are several tools that can be used to get that. So as soon as you get your phage genome, you first of all want to blast it to check, is it a new species? Is it a new genus or whatsoever it is? But over the years, the ICTV has also found and classified the fact, getting a main criterion for classification of virus to show that within any virus species that is less than 5, that vary less than 5% at nucleotide level, belong to the same species. So, and it's an important tool that can be used to do this is a Viridic. So you could read up more by Christiana group in Germany. They developed a fantastic tool. So this tool worked along with the BLAST tool for you to identify if you have to classify your viruses. So for Pseudomonas vesicola, so what was done is we isolated the virus and then extracted the DNA, performed an assembly, conducted the blast in the NCBI. And when you do a blast, you see a number of species that are similar to it. So what we did was to pull all of those together and then performed a similarity profile on Viridic for classification. So one of the things you see is the Viridic results showing us comparing Pseudomonas vesicola with quite a number of phages. So one of the things you notice that phage vesicola here shares about 97% with another phage that belongs to a myoviridae. So and the myoviridae myoviridae are not quite much. This is about the 27th virus in the myoviridae group. So you have such that it's within 95%. So it belongs to the myoviridae. And so some of the tools to use for you to in your phage classification and phage study is so spades for assembly. You trim the adapter using we trim using Numeracy. We have found Patrick to be very interesting. Patrick was used for annotation. It brings together quite a number of tools. So for those people that are not very conversant with bioinformatics or the back end, because that can be very cumbersome, Patrick can solve the problem of trimming, and it also complements Galaxy in a way. So we also use FACT for life cycle of phage. So most of these they have their APIs and it's quite easy. And the Viridic, so the Viridic, although it has a little bit of, but it doesn't work with the back end. You just assemble all the genomes together and they still work. So one interesting part for this is that you don't need sophisticated skills to be able to arrive at this. So basically, to name our virus, one of the things we basically did, been able to isolate, but this study forms part of a larger study. It's actually a study towards, we are currently evaluating Pseudomonas aeruginosa phages for the treatment of, as an alternative for the treatment of diabetic wound. And also one of my PhD students is working on Salmonella phages as an alternative for management of Salmonella typhi. So these are some of the tools we use for classification and naming our phages. So Nagode, it's in Igbo, in Hausa. Thank you for listening.
Thank you very much. I think we have room for one question. Any burning question?
Just to ask Nemeka, thank you so much. Samuel, I'm already here. Yeah. Thank you so much for this. Just for the purpose of clarification, was whole genome sequencing performed on this Zikora?
Yes, that's actually the first thing to do for you to. Yes, it was performed. That's
okay.
Because
one,
for you, yes,
there is a follow-up there because then in terms of the tools that he used, because it was not clear to me, if I understand, the Patrick it's already like an online tool where you can submit the whole genome and maybe perform the analysis with already, a platform like Galaxy, something like this? Or was there a command line like, the normal workflow for the whole genome sequences analysis?
Okay, so what we did was I used the command line for spades in my computer and also used the command line for Trigonometric. So I used Patrick only for annotation. If you decide to use Patrick for annotation or for assembly, you could use that, but I use Patrick for annotation. Basically, yes.
Yeah, finally from my end, I was just thinking, do you think that if eventually phage is embraced as an alternative maybe to circumvent the current challenge we are having with antibiotic resistance and drug resistances, do you think that this phage will not misbehave in the course of time and there will be mutation and the virulence might set in vivo?
I think that question is a question everybody naturally should be afraid of because of COVID and what viruses are doing now. But phages are specific for prokaryotes. They are specific for bacterium. And although we have macrophages also against viruses, fungi, it does not have any effect against, over the years, clinical trials have shown that it does not have any effect against, it doesn't have affect any human cell. So the risk of mutation into becoming a virulent pathogen, it's minimal. And I don't think it's something that would come up at any time.
Okay, thank you.
Very much. Can you stop sharing, please? So straight away we go to the next presenter in the person of Dr. Norbert Nwankwo. So Dr. Norbert graduated from the Faculty of Pharmacy in the University of Nigeria, Nsukka. His interest and passion for bioinformatics started during his last year at UNN after being taught by Professor Maurice Iwu that computer could be used to design medication. Dr. Norbert submitted research proposal for a PhD in bioinformatics to the Montfort University, Leicester, United Kingdom. He was later admitted in 2012. He completed his program at that university and engaged to digital signal processing technique to investigate biological characteristics with emphasis on HIV/AIDS and malaria. This led to the discovery of novel approach that now computationally calculates and numerically present biological features rather than measure them. Dr. Norbert also invented 5 biomedical devices, including computer-aided drug resistance calculator. This is a rational point-of-care device that quickly determines the most effective medication for ailments. This is to help the physician with results necessary for informed decision on their patient. Upon returning to Nigeria, he has been teaching pharmacoinformatics and research methods at Madonna University and also at the University of Port Harcourt. He has recently returned to the US where he continued to teach. And in recent times, he has learned that this is easier to invent than to maintain a grip on your own invention. Welcome, Dr. Norbert, you have the floor.
Yeah, can I share my slide?
Yes, please.
Okay. Yeah, I believe it's visible to everybody.
Yes, it is. Thank you.
Okay. Let me quickly. Oh yeah. Yeah. Thank you very much, Ademola, for the introduction. In a couple of minutes, we'll be looking at the reliability of these devices we generate from both dry lab and the wet lab. I will be putting across to you the confirmation that our devices and the technique we had here were, first of all, authenticated by the use of wet lab and dry lab procedures. As we all know, and right now as we talk, we are aware that we have two devices that are unmanned about 55 million kilometers away in a planet called Mars, where they are investigating stability of life. And it's interesting to let you know that in 3 months alone, we had about 17,000 pictures sent from the Mars to the Earth. All this could only be achieved because we had reconciliations both from the physical experiments and the computerized experiments. As we all know, all fields had devices that are computerized, including pharmacy, and like I said, I will be presenting a reconciliation we did. But before I do that, I will put you through with what digital signal processing techniques involved. And I'll also be putting you through us with the device we discovered. As a matter of fact, we got the findings or the outcome from this biomedical device and reconciled it with already existing or already documented outcomes. Apart from that, we looked into other works done by other scientists, and we found out that there is a correlation, particularly a linear correlation between the results obtained at the wet lab and those obtained using dry lab. I want to start this introduction by saying that all bioactive molecules, including medications, are known, which might be in nature alkaloids, terpenes, it could be saponogens, and no matter what it is, and no matter what the structure is, are known to have target proteins. I'm happy we just had one of the examples is talking about multi-drug resistance, falciparum. These are genes that are encoding the same bioactive molecules. And naturally, we have some bioactive molecules that are protein in nature. We have insulin, for example. And the good thing here is that if you're able to assess the biological characteristics of these genes, proteins, you also get back to the biological functionalities of these active molecules, bioactive molecules. We had ascertained that that was one of the work I did during my PhD program. We took two bioactive molecules. These are purely antiretroviral. One was a bioengineering product of the other, and we assessed the biological functionality in terms of efficacy using a procedure which is biophysical. It was found that the second antiretroviral has more activity over the other. And when we use this particular digital signal processing techniques, we also found out that the result correlated. And as we all know, proteins and peptides are amino acid in linear formation. And as we speak, we have more than 540 amino acid scales, which we also call the molecular descriptor. Researchers like me and you had done work in various interactions that involve protein. And we know we have 20 essential amino acids. They were able to ascertain the level of interaction provided by each amino acid and put this in the database, which they call amino acid scales. For example, we have a binding interaction, which is called electron-ion interaction potential. In this, we have various amino acids like alanine presenting their level of interaction, their level of binding. When we have this together and we apply digital signal processing techniques on these proteins, we will be able to get back to the biological functionalities that is manifested by the same agent. As a matter of fact, we have been able to work on over 1,000 proteins using this particular method. I might want to start by going through what the procedure entails, and I will be using these two little peptides. I had used these slides before, and the peptides are very, very small, so it's easier to explain with these peptides. You have peptide 1 and 2, and the first thing we do is to translate the alphabetic codes into the numerical sequence. We have a sequence of alphabets. What we then do is to use the values we have from the amino acid scales and interchange them. And what we get is numerical sequence, and that is what we call signal. And the next thing we do is to apply discrete Fourier transform on this sequence. And what you get is a plot with various peaks. This procedure is what we call informational spectrum method. Individual plots are regarded as informational spectrum, while point-wise multiplication that gives you a common position of interaction is what we call common informational spectrum. This is an example of one amino acid scale or molecular descriptor. And in this case, like I said, this is for binding interaction, like I said, electron-ion interaction potential. If you look at this, you see A represented by 0.373. A is alanine. I here is the isoleucine that has 0, and also leucine, L has 0.2. The last one is valine, the value is there. We have over 540 of similar table, including structural activities, structural properties, helix, alpha, folding. I remember we had a guest talking about mixed folding as it associates to mad cow disease. If I'm to investigate that, which might be the next work I'll do, I will concentrate more on folding properties. But that doesn't mean that you shouldn't bring in the other properties in order to ascertain or obtain a complete result. Now, if I put this, if I interchange the sequence, the first sequence I have here, VIP and the rest, with the values of binding electron-ion interaction potential, we'll have these values here. And if I plot that, I'll just get that, which is a signal. The second peptide is this, and I will get another signal. Now, if I apply digital signal processing technique on this, I'll have amplitudes on these positions. And as a matter of fact, to find out a common point of interaction and the contribution by each of the peptide at various points, you do what you call pointwise multiplication. Obviously, you have a plot like this. The first one is a mirror image of the other. And normally you don't need the other part. So that's why I have frequency B. So this is the nature of the informational spectrum provided by that peptide. Now, the first figure here is for peptide 1. The second one is for peptide 2. And look, the third figure gives you the pointwise multiplication. And that is a common information spectrum. If you look at position 2, that's where they have maximum point of interaction when you combine the two. And if you look at the first and the second plots, you discover that at position 2, they have reasonable high level of activity. Now, this procedure, I tried to explain this procedure with that little peptide. What we did in an attempt in this discovery that the computer-aided draw design calculator is that there is a work that is done already by some Danish researchers where they correlated outcome from the IC50, which you know is a wet lab results obtained from various mutants. They engaged HIV protease enzyme that is the target for Ampreneval. They came up with results. I decided to apply this procedure on the results they had. The first plot here is the sequence itself as obtained from Stanford database or UniProt. I got it from one of them. Then you will notice N and K as the point of mutation. I chose a random sequence in order to help validate the results. The B is the result of a plot which represents informational spectrum of one of the sequences. We had about, we generated over 1,000 plots, and I just chose some of them in the submission I made to USPTO. Now, in this slide, A is the consensus position of interaction. And using that position, we're able to determine the level of interaction provided by this mutant, and you see it's 0.22, which represents 71%. Now, this level of susceptibility or result were obtained, and we decided to look at those provided by that work, and we found there is a correlation. We made this submission, and in the course of this submission, I had a paper sent to me by the patent attorney because I think the examiner was looking for more correlation. So I found out that researchers had also done similar work using different methods, in this case, computational scanning. I was shocked to find out that similar result was obtained with what we had here. A linear correlation was obtained. I am very much happy to say that with these correlations we had, it becomes possible to attest to the authenticity of the procedure vis-a-vis the reliability of the device. And I'm happy to tell you, 3 weeks ago, we got an allowance for the patent. Apart from this, we had other devices that we got from this procedure. The inventor of this particular procedure, they are from Serbia, and one is in Australia. Irena Kossi had already provided this tool is a free tool for phylogenetic analysis. All you need to do is to put several sequences there and they give you the tree. When I came back, shortly after I came back to Nigeria, I had a company over here in US that is looking out for a computational procedure to assess vaccines. I think we're about 184 that vie for it globally, and mine was accepted as the solver. So we have a computer-aided vaccine potency assessor now. Recently, too, we had another one that takes care of drugability, in which case you don't even need to undertake all those routine procedures that entail using materials, maybe some time, to ascertain which of the candidates of drug or vaccine are druggable. We also have another device that takes care of potency of drug. Recently, I also looked at the procedures we have in the lab. Some of us here had I remember those days we were using a guinea pig ileum to determine antihistamic activities. I found out that the same information we are seeking from there could be obtained using the sequence information we have by analyzing them using this digital signal processing technique. I would want to say that we all know the value of AI, and like I mentioned, without AI, we cannot be studying the planet Mars with an eye to say live there. We're expecting them to find out whether we have water there, which is intended to be split into hydrogen and oxygen so that some of us will move over there before there will be, Armageddon, if there should be. The reliability we obtained here had helped authenticate the usefulness of the device we got. And I'm also glad to let you know that nature made it possible. And we have these bioactive agents being associated with protein targets and genes, which makes them analyzable, and also the ability to unveil the biological functions embedded in them, which might require a lot of resources if we have to look into the various nature. If you're looking at alkaloids and the structure even is frightening. I also want to say that one good thing about this procedure is that it engages Fourier transform, which we know is the foundation of technologies like radar speech processing technique. We know what radar is. Without radar, we can't be keeping thousands of human beings on the air.
Sorry to interrupt you.
Yeah.
I think you're out of time.
Okay. So finally, I want to say that I'm glad to share with you the device, the procedure, and I want to let you know that despite the pains this discovery has cost me, I'm happy to let you know that this belongs to one of you. Thank you so much.
Thank you very much. This is really interesting. But have you in any way, I just want to ask a quick question. This device, is it been deployed in any way, commercially?
Yeah, we were just through with the patent. Like I said, we got the allowance 3 weeks ago. So the next thing now is to develop it, and collaboration, investors. We're talking with investors, and if there will be investors from over there, that will be fine. But we are discussing with investors now as to the development of the device.
Great, awesome. We are running behind time, and we would like to change our gear and move a bit faster, and which I'm not sure we would accommodate more questions at this point, unless if there is one very, very burning question that you can't. So I don't see any question in the chat, and so I think we will move on. Thank you very much, Dr. Norbert. This was an excellent presentation, and I wish you all the best in deploying and developing this device. Yeah. So straight away, we would move into our next presentation, and we have Dr. CG Anauko. I'm not sure if I pronounced that very well at all. I'm sorry if I murder your name, but please pardon me. She will be presenting on comparative metagenomic analysis of crude oil polluted and unpolluted soil in Okakri, Ahoada local government in Rivers State, Nigeria. So, Dr. Anauko, please, you have the floor. I can't find you. Oh, yes, I do. Yes, please, you can share your screen now.
Yeah, thank you, Toyin.
Yes.
I don't know, can you see my screen?
No, we cannot.
Okay.
But we can hear you.
Okay.
Yes, we can see your screen now. Thank you.
Okay, thank you, Toyin. And thanks to NGBC for this opportunity to share a part of what I've done with bioinformatics. Actually, I'm still a learner in bioinformatics thanks to MRFSS. Yeah, I'll be presenting on the
full,
Yeah.
Yeah.
Okay.
You. Yeah. Thank you.
I'll be presenting on the topic you already stated, comparative metagenomic analysis of crude oil polluted and unpolluted soil. Already my name, and you can see my affiliation, Department of Applied Microbiology, Nnamdi Azikiwe University. By introduction, yeah, we know crude oil pollution of the soil, the lithosphere, has become a serious environmental problem since past decades, especially here in Nigeria. It impacts heavily on soil structure, microbial diversity, and activities of soil organisms. That is a typical activity, and what it needs here is
Hello? I guess she's having a network problem, I think. Maybe we'll wait for a few seconds and see.
While we wait for her, just a brief announcement on what is left for us today. Immediately, Dr. Nugu is done with the comparative metagenomic studies. Please, we have all important seminar for everyone, the undergraduates and all of that, on starting your bioinformatics career. And I think Professor ICC is already on board. Then after that, we take a Nejo. And I think we have 4 poster presentations for 5 minutes each, then before we end the day. So still, let's hope that by 1 o'clock, we shall leave here. So thank you.
Sorry about that. It's network issue around here. Yeah, the aim of the study actually, for the purpose of this presentation, is just to investigate the impact of the long-term crude oil contamination of soil on the microbial community diversity and abundance, comparing it with an unpolluted soil. Then the methodology I used first is to collect the soil sample. So I collected crude oil polluted and a pristine soil sample from a polluted soil and unpolluted soil. And the sampling site is a local refinery somewhere in Nigeria, in Rivers State actually. Then we did genomic DNA extraction. I used the QM Power Soil DNA kits and processed on a QICube automated nucleic acid extraction system. Then deep sequencing of 16S rRNA gene amplicon was done. The primers used were CS1515 forward and CS2826 reverse, targeting the V4 region. Then PCR condition as shown were used. Then the libraries that were recovered were pooled and purified, then went in and then loaded onto Illumina MySeq for sequencing. The reads that were obtained were now analyzed.
Sorry, Chidekondle, your slides are not moving. Could you move the slides? I mean, it's just, we're still looking at the end.
Wow. I don't know, from my end, the slides are already moving.
Moving. Okay, go ahead, go ahead. We don't have time.
Okay. But let me know if I can resolve what the problem is.
Yeah, no, it's Nigeria issue. Just go ahead.
Okay, the bioinformatic analysis, I analyzed the reads that were obtained. I used the PS software package. Yeah, the merged reads were trimmed, chimeric sequences were identified and removed. Then using use search algorithm compared to reference sequence database, then taxonomic annotation and sequence abundance data were then merged into a single sequence chart to give summary of absolute abundances of taxa for all phyla, classes, orders, families, and genera. I don't know if my slide is still showing, because I want to go into the results section.
Yeah, it's still showing us aim.
Wow. Please, let me try to resolve it.
It's still showing the aims. Can you try going on a full slideshow? Let's see if that will work.
Okay.
You need to go up and click slideshow beside animation instead of clicking it from the bottom. I think you have an old version of PowerPoint.
Am I there?
Yes.
Yeah.
Okay. Let me go through the results.
It's better now.
Okay. Going through the results, the metagenomic analysis revealed the presence of large communities of microorganisms in both the crude oil polluted and unpolluted soil. And the Illumina MySeq sequencing showed that there were about 47,006 and 53,069 OTUs in the polluted and unpolluted soil samples, respectively. Now, this is the percentage abundance from the soil samples. We recovered both archaea and bacteria. And we can see that the bacteria were more in abundance than the archaea. Then also this showed the relative abundance of the archaea phyla in the soil samples, and I recovered more of the eukaryota in the polluted soil than in the control soil. The reverse is seen with eukaryota. Then I tried to show the relative abundance of the bacterial phyla in the soil samples, and we can see the soil samples, the distributions of the various bacterial phyla in both soils. While the actinobacteria was more in abundance in the polluted soil, the firmicutes were more in abundance in the unpolluted soil. Also, you can observe here that a greater percentage of the reeds that were recovered were not classified. And this is as a result of the database that was used. Yeah. Then this is the relative abundance of the bacterial classes for both the soil samples. The same, we discovered that a greater percentage of the reeds were not classified. They were not identified. The same for the figure 5 here is the relative abundance of the bacterial orders. Several bacterial orders were recovered in the soil samples, while the actinomycetes were more in the polluted soil. The bacillus were more in the unpolluted soil. Then we also looked at the families of bacteria in the soil samples as shown in this figure. Then I'll run through it, the abundance of the bacterial genera in the soil samples. There were more bacillus in the unpolluted soil, while there were more Candidatus corybacter genus in the polluted soil. Of course, you can see in the unpolluted soil, almost 50% were not classified. Yeah, I still relate it to the database that was used. Then this is the rarefaction curve of annotated richness. Yeah, this curve, it is a plot of the total number of distinct species annotation as a function of the number of sequences that were sampled. Now, if you look at the left side, there is a steep slope. This indicates that a large fraction of the species diversity remain unidentified, as you can also attest to it from the other figures I showed you, where a greater percentage were not actually identified. And we owe it to the database that I used. Now, this is the alpha diversity values of the metagenome. This is showing the population of species that were actually recovered or discovered. For the polluted soil, up to 41 species, while unpolluted soil, about 36 species. Now, the arrow is showing the position of the alpha diversity for each of the soil samples. Now, this is a sequence length histogram. This shows the distribution of the sequence length in base pairs. And if you look at it, the sequences that were obtained in the various soils fell in from 250 and 255 plus. That is the length of the sequences that were obtained from the soil samples. Now, I'll conclude here. Like I said, this is just to show the microbial diversity and their abundances. And the community of organisms recovered, especially in the polluted soil, going from the archaea through the bacteria, are actually reportedly hydrocarbon degraders. The main focus in the research is to see if the indigenous organisms can be utilized to carry out reclamation of the soil. That's why it's very important for us to understand the community of organisms in the soil before you can employ any bioremediation technique. Now, they have been classified as shown in the results into their respective phyla, families, classes, and genera. And further analysis is still ongoing on the sequence rates to establish their functional genes and metabolic pathways for application in bioremediation. Thank you for your time.
Thank you very much. Awesome presentation. Yes, we have a question from.
Yeah.
Good.
Thank you, Chikwadele, for this. Yeah. So I think you have difficulty to get to the species of this, probably because of the type of platform you use. This is 16x. Sometimes it's difficult to get to the species with this, but maybe with shotgun, it might be a bit possible. Then again, let me make it as a proposal, not a question to you, but if you could, please, in the course of the further analysis, look at the what we call the alpha beta diversity and the alpha and beta diversity within the polluted, and also beta comparing the two. So because within each sample, there could be evenness and also there could be uniqueness within. Then also you could now look at the two samples. So we have also a script that can be able to run this on that Data2 platform.
Okay,
Yeah.
thank you, Dr. Moses.
Yeah.
Someone is asking a question from the chat. I was asking, which database did you use for this analysis?
Yes, I used Green Gene.
Okay. Yeah, I wanted to ask, what pipeline did you use for your analysis again? I'm not sure I really got that.
Use search algorithm.
Oh, okay. Good. Awesome. So thank you very much for this wonderful presentation. And then we would go straight away to our seminar. And if you are interested to start your bioinformatics career, and then this is something you have to look forward to. So we would, the presentation will be done by Professor I.C. Uzochukwu, who would guide us through how you can start your bioinformatics career. Prof, I think you should be able to. Yes, you have the floor. Thank you very much.
Okay, good afternoon.
Maybe we'll ask Prof to make the
I hope my screen is showing.
Yeah, but you need to make it full.
Yeah, I will.
Okay.
Currently as the Director of Research and Innovations, group called Drug Design and Informatics Group. I'm very happy to be here to talk about this, starting your bioinformatics career, because this is something that I'm quite passionate about. You know, how do we spread the knowledge about bioinformatics in Nigeria plus Southeast? And a lot of people may be shy, you know, as per the initial inertia. You know, you'll be in microbiology, you talk about the lag phase of microbial growth. So how do we really start? So I want to get through this slide in a short while. We say, what is bioinformatics? We look at the scope. Sorry, I was not here throughout. I'm just coming into the country. And so I've not been following to know exactly the scope of the discussion, but I know that a lot of things has been covered. So I will look at the scope briefly, look at applications, then now go to the how to really start the career. So I say define yourself, place a niche, submit to mentorship, let your light so shine, build your network, requirements, what a bioinformatics lab should look like, then we conclude. So this is an overview of what bioinformatics is. Basically, when you bring biology and marry it with these three areas, mathematics, applied mathematics, computer science, and statistics. So you are trying to apply biological data. You are trying to apply computer science to biological data, or you're using computer technology to store, analyze, and interpret biological data. So you see a lot of things are involved: the chemistry, the biology, the biotechnology, the genomics, the statistics, biochemistry, all kinds of stuff. So basically, there are two broad areas. It's like the two sides of a coin. So we have the computational biology angle, which has to do more about with the development of tools, you know, development of databases, software, etc. Now, the other side of the coin is now the application of the tools and databases to generate new knowledge in biology and biomedical research. So we are not going to run through all these applications, but these are just some. drug targets. You want to find the drug targets, giving large database, of a genome. You want to discover new drugs or design new drugs, plant taxonomy, phylogenetic relationship, even understanding biochemical processes, identifying your biomarkers, forensic DNA analysis, agri-biotechnology, personalized medicine, virtual high throughput screening, sequence analysis, homology modeling, similarity searches, drug lead optimization, physicochemical modeling, even drug bioavailability, and so on and so forth. So the applications are diverse. Once you have the skills and you have the tools, there are a lot you can do. So we now get down to what do we do to start. So the key thing is to define yourself. A philosopher said, man, know yourself. You need to know who you are. How do you approach that? First of all, what did you study first degree? That's my approach to it. If you studied pharmacy, if you studied medicine, you studied computer science, you studied microbiology. So you need to know who you are, because that's your area of strength. So you define yourself, what kind of challenge do you usually want? Then you also define your place and niche. So you need to carve a place for yourself. You don't need to be me. Be yourself, define your place. So how do we do that? You look at your research problem. Which research problem interests you? And then when you identify that, you try to build your skill, bioinformatics skill, around those research problems. Because there are a lot of things to really know. There are a lot of things to know. There are a lot of things to study. There are a lot of things to pursue. But you cannot pursue all of them. Bioinformatics is a big elephant. You can only eat a part of it. So when you have defined your place, your niche, you try to upskill in that chosen path. You develop skills, attend courses, attend conferences, attend workshops, read a lot in that area. So I'm just giving idea of some of the things we can do. You can go to, all these online courses, and of course, and read up a lot of things. Go to databases, read up a lot of things, and so on and so forth. Then you try to publish in that area, because that is what will also define you. And then if you're looking for funding, look for funding in that area. That will also, you are building your capacity through in that particular area. So, and then you try to collaborate. Don't be don't do a one-man show. Don't do a silo research or be on your own. You need to really learn from others. And the more you collaborate in a multidisciplinary manner, the better for you. So you collaborate with people from physical sciences, people from health sciences, people from engineering, people from geology. So you collaborate, so that you also be able to interface what you are learning with other aspects of human challenges. So one of the key things about growing as a starter, as a young person, is mentorship. So you need to submit to mentorship. The advantage is that you're going to stand on shoulders of people who have gone ahead of you in that particular area, generally, and in your own area. So, so you need to find your mentors, a mentor or mentors. So you must not have just one mentor, but you may have one principal mentor and then find other mentors. Then you try to wash your mentor's feet. Very important. You carry their handbag, you walk around them, take assignments from them. Then when they give you assignment, you take it seriously. What do we mean by that? Diligence, deliver diligently. Timeliness, most things have timelines, so you try to beat the timeline, so they will give you more. They will trust you. And then there has to be honesty, both in your research integrity and all that. And then that will make a place for you in the heart of your mentor. Then as you learn, you also teach others. You learn by teaching also. So you learn, you share your knowledge, teach others, and in that way you grow. So I just have this something from mentoring. So your mentor is going to coach you, give you advice, motivate you, give you direction, and also share goals or set goals with you and support you along the line. So as you grow, you let your light so shine. You know, in Matthew 5:16, say, let your light so shine before men that they may see your good works and glorify your Father in heaven. So to do that, you need to position yourself, become visible. You need to have social media, you have personal blog, you volunteer in assignments like we have a workshop, you volunteer to do this or do that, you know. Then you do what I call adjacent possible. You expose yourself to new environments, new circumstances, new fields. In that way, new things will open up to you, new horizons, you know, new challenges will open up to you. So explore new challenges, go new places, interact with new sets of people. In that way, a lot of things can begin to happen. Then build your network. Remember to build your network. You have your alumni peer group, you know, people you graduate with or you're working with, colleagues, senior and junior, Facebook, Twitter, all kinds of things. Build your network so that you can gain maximally from your skills and the opportunities. Basically, what do you require? Yeah, because the resources required to start bioinformatics is minimal, really. You can start with your laptop, and that's a lot of us do have. Of course, you need your brain power, the ability. In fact, the greatest gift that God has given man is the brain. So you need to deploy it. Then you need internet access, because you may need to access certain things, download certain things, even if you're going to use them offline. And then you need software, and many of them are open source, many of them are free. Then you need also primary data. You know, you can, a lot of places where you get primary data to do your further studies or analysis. And then as you grow, you upgrade your systems, upgrade your computer, you know, upgrade maybe from laptop to desktop, from desktop to high performance computer, upgrade your internet access and all that. So you keep upgrading until you have a system that matches your capacity or your desire. So what does a bioinformatics laboratory look like? So this is just a picture of what we have in our university in the drug design and informatics laboratory. You know, you can see you're going to have some desktops, so you can actually set them up, maybe have a printer, have a conference table, and then have a projector so you can project and discuss issues and all that. If you look here, you also need to see that you need a power supply, especially for those of us in resource-poor countries where electric power may be minimal. So you may have to install inverter that can store power for some time, so that maybe if you're doing a virtual screening or simulation, you can set them up and go home. And because you have continuous power supply, you can come back and take your results. So these are some of the things you may need as you grow. So let's conclude. We're saying you need to groom yourself in the area, network globally, scale your potentials, and then wait for the returns that will come. Thank you, and God bless you.
Thank you very much, Prof. Maybe we'll give room for very few questions because we are fast running out of time. Okay, yeah, Dr. Moses.
So I think I wanted to clap for Professor before my hand went raising, you know. Just to say thank you, Professor, for the presentation. And maybe in the course of this presentation, I could go down the memory line when we all started this definition of bioinformatics. And the key thing that entered my heart was, as you learn, you teach others. So I think I've been doing my best to teach others from the ones I've learned.
Exactly.
Yeah. Yeah. So this has really helped somehow. And also the passion around the whole thing, because without passion, it's going to die. Yeah. Thank you so much.
Thank you.
Thank you very much. If we don't have any further questions, then we can move straight away to the poster presentation. No, before the poster presentation, sorry, we have one more speaker, one more presentation. And I think, if I'm not mistaken, we have Ocho. I have a difficulty pronouncing this name. Ocho Kenemi Enejo. Sorry. Yeah. So this presentation is on bioactive compounds of Anona. Senegalese peers exhibits hepatoproactive and anti-clastogenic activities to counteract sodium arsenite-induced toxicity. It's an in vivo in silico studies. So you have the floor, Dr. Ojo Chenim Energy. Sorry for the pronunciation. Yes, we can hear you.
It's all right. It's Ojo Chenemi.
Okay. Sorry.
Okay, can you see my slide in full screen?
So, yes, we can see it.
Okay, thank you. Thank you very much. Good afternoon, everybody. I'm delighted to be here, and I want to say thank you to the organizers for allowing me to do this presentation. My presentation is entitled Bioactive Compounds of Anona senegalensis pears exhibit hepatoprotective and anti-clastogenic activities to counteract sodium arsenite-induced toxicity. We did both in vivo and in silico studies. I'll be guided by this outline during the course of my presentation. Now, arsenic is a ubiquitous, naturally occurring element, chemical element, which is implicated in disorders such as skin lesions, peripheral neuropathy, cardiovascular diseases, and cancer. And the major way which humans are exposed to this toxicant is through consumption of contaminated drinking water. And this has posed a major public health concern. It is said that oxidative stress is considered the central mechanism through which arsenic exerts its toxicity. And this is fundamental to the progression of hepatocellular cancer, which interact with biomolecules and damage cellular components such as lipids, proteins, DNA, and possible apoptosis of hepatocytes. Now, natural products have formed the basis for the treatment of human diseases. And in fact, there's a lot of folkloric information for various plants which are used to treat different diseases. The one that was used in this study is Anona senegalensis, which is part of the Anona family. It's commonly called African custard apple, and then locally it's called Gwandan daji in Hausa, Abo in Yoruba, and Uburo Ocha in Igbo. And the plant parts have been reported to be used traditionally in treatment of various ailments. Studies also on the plant have revealed that it has analgesic, antibacterial, and anti-inflammatory activities. Despite its wide ethnobotanical uses, especially in relation to cancer, compounds identified in this plant are yet to be matched with specific biological activities, especially those found in the leaves. At this point, I'd like to mention that while I was working on this plant, a colleague of mine confirmed that someone used this plant in the north, and it actually treated, liver cancer. But we really don't have scientific backing, to corroborate that. So that was also one of the reasons why this study was undertaken. And so what we did was we collected plants from Erua in Ibadan, dried them. They were identified in the herbarium, the botany department, University of Ibadan. Leaves were dried for about 2 weeks and then extracted for about 72 hours using distilled water. We also carried out quantitative phytochemical analysis on the crude extract. And the study design involved 200 milligram, administering 200 milligram per kilogram body weight of this extract to rats that had already, received sodium arsenite, which is a toxicant. And so at the end of the assay, it was just a toxicology study. So the assay lasted for about 2 weeks. That's like 14 days. At the end of the assay, at the end of the experiment, we carried out assays such as ALT, AST, MDA. There were actually several assays we carried out, but the data is so much. So I just chose a few, to present here. We also did a micronucleus assay to test for clastogenicity. And we did histopathology. And I mentioned that from this title, that there are 2 parts to the study, in vivo and in silico. Now, based on the results we got from the in vivo studies, we went further to try and understand, what was really going on at the molecular level. So we employed bioinformatics to do this. So what we did was we downloaded ligands, which have already been characterized from these plants from the PubChem database. And we prepared them using the league prep module on Surginja suit. And proteins implicated in apoptosis, cell proliferation, and redox, sensitive pathways were select proteins, because there are so many. So select proteins were downloaded from the PDB database, and then were also prepared using the protein preparation wizard module on Maestro. And glide standard precision and extra precision protocols were performed using glide still on the Schrodinger suit. We also did ADMET analysis, and this was done using QuickProp. And at the end of the day, our lead compounds were screened using the Lipinski's rule of 5, which states that a compound should have a molecular weight of less than or equal to 500 grams per mole, should not have more than 5 hydrogen bond donors, and 10 hydrogen bond acceptors, and log P should be less than 5. So these were the criteria we used to choose our lead compounds. Okay, so from the phytochemical analysis, we see that there's an abundance of alkaloids, flavonoids, and carotenoids. So this, this tells us that the plant could be therapeutic. Well, how is what we were working on to find. And the liver enzymes, were significantly increased in the sodium arsenite group. But after administration, this was brought down to as low as control levels. So that tells us that the plant is working. And then MDA levels were also tested for. We can see highly elevated MDA levels in the sodium arsenite group. However, sodium arsenite and Anoda tegalensis extra group was greatly reduced, down even almost to control, and the sodium arsenite only group. For the micronucleus assay, we can also see that sodium arsenite caused a lot of aberrations. This is shown by the high number of micronucleated formation from micronucleated cells in the bone marrow of the rats. However, this is reversed after treatment with the extract. Histopathology also shows us that the extract was able to reverse the effect of sodium arsenite, as revealed in group D, where we see only mild periportal cellular infiltration compared to severe lesions in group B, which is a sodium arsenite positive control group. Now, for the computational studies, this is a heat map showing, I mentioned earlier that we got some compounds. So after downloading the compounds, they were docked in the active sites of the various proteins. Now we can see some red cells. These are the cells that reveal very tight binding of the ligands to the proteins. We can also see yellow cells and a lot of green. So it shows us that the extract is actually working, but the focus here was on the tight binding proteins, or the tightly bound molecules to the proteins, where we have 4KIK, which is IKB, you know, that has a lot of binding to this compound. These compounds, anoline, assimilibone, catechin, cochlaurin, isoboldine, nonanentine, non-luciferin, noroliverolone. They are all alkaloids. And it corroborates the results from the table that showed us the phytochemical analysis, where we had, you know, quite an amount of alkaloids in the plants. Catechin too bound, you know, showed good binding to most of the proteins, which could suggest that catechin, being a flavonoid or in the class of flavonoid, suggests that the plant had exerted its effect maybe via antioxidant mechanisms. Okay, here we see some binding of the ligands to IKB. And we can see a lot of, for most of the, in fact, all the compounds have green patches. That shows hydrophobic bonding. And in molecular docking, hydrophobic bonding is important. And we also see some hydrogen bonds. So 2 bonds that are important, hydrophobic bonding and the hydrogen bonds. So we can see them represented by the green, and then the arrows, purple arrows on some of the distance. So this bonding, hydrophobic bonding, and either hydrogen bond, ligand either donating or accepting hydrogen bonds could serve as a reason why, you know, they have tight binding to the proteins, these ligands. Here, so we also see that JNK binds well with some of these compounds. Now, this result was extracted from the heat map I showed you earlier, the ones that had tight binding that were represented by the red cells. I had to get their two-dimensional binding at the active site so that you can see what is actually going on. So you also see that hydrophobic bonds are in abundance as well in this case. And NF kappa B also, which is a transitional element, also has good binding to this, or the ligands have good binding to these proteins by the representation of the hydrophobic bonds and the hydrogen bond. The quick prop results show that all the lead compounds are druggable, because, as I said earlier, the molecular weight should be less than or equal to 500 gram per mole. And in fact, the highest we have here is just about 327. So it ranges between 200 and 300, which are all less than 500. Hydrogen bond donor is also within the range. The hydrogen bond acceptor, that's its next column, HBC, accept HB, is also within range. And then we can see the last column that says RO5 violation. None of these compounds violated the rule of 5. So this makes them good candidates for further development into drugs. Okay, here I just tried to show us a plausible mechanism or mode of action through which anona-salicylensis, you know, is exerting its effect. So we can see from the results, it was able to inhibit IKB quite well. NF kappa B and JNK. So I am just giving us this as a plausible mode of action through which it is working. The
Out of
results
time.
clearly showed that sodium arsenide caused significant damage to hepatocytes, as revealed by the results that were displayed. Okay, and this is my, but we can see that all of these were ameliorated after an annona-senegalese extract administration. And so it's safe to say that the anti-inflammatory and antioxidant activities of compounds present in this plant may have accounted for its hepatoprotective and anti-classogenic or anti-cancer properties. Thank you very much.
Thank you very much.
Wonderful.
Okay.
I'm not sure if we have any questions.
Dr. Toyin.
Yes, can you hear me? Yes, good. Thank you very much.
Yes, I can
do
hear
we
you.
have any question? I don't see any question in the chat.
So is there any plan to formulate the Annona senegalensis, which, I mean, I think I have a publication on that plant, which is very has great potential. Is there any plan to formulate this Annona, probably like a tea, or should we continue recycling research around the Annona senegal? Can we do something to maybe push this into, not really in the market, but so that people could know that they can even use this as a tea?
Well, we can work together to do that, because based on, it's true now, from these results, we have seen that, it has great potential. So it's something I have not really thought of it.
Yeah, you need to think when Artemisia annua came in just like this. Well, Artemisia annua is what gave us the ACT we are taking today. So I think that if maybe more collaborations are sought, then there could be, people could come into this and we can use Annona. Just people drink Annona, to be able to control their health issues.
Yes, that's good enough. Thank you, Mr.
Yeah, we have
Dr.
one
Moses.
question in the chat. It says, what is the medium that you use for the extraction?
Medium.
Yeah, I'm not sure.
Yes, I'm not sure too, but it is aqueous. I use distilled water, so it gave me aqueous extract. I don't know if that answers your question, but it was distilled water I used. The leaves were soaked, pulverized leaves were soaked in distilled water for 72 hours and subsequently concentrated.
Yeah, that is more reason why it's going to be a tea. If aqueous is working this way, then it's nice, because when you use methanol, use ethyl acetate, at the end of the day, these are not what human beings could consume.
Yes,
and
actually,
the
study was 2 parts. I also did the methanol extract, compared it with the water extract, but the aqueous extract was better than that of the methanol extract.
More reasons to make it a tea. Okay.
Yes. So I'm open to collaboration.
Okay. Thank you.
Yes.
Thank you.
Thank you
Thank
very
you
much.
very much.
So we would move straight away to the poster presentation. However, I will hand over now to Dr. Moses to push this ahead. Thank you very much.
Yeah, thank you, Tony. Guys, you may know that Tony is like after 11 p.m. in Australia, helping us to hold on to this section. Now he might need to sleep so that he will attend church tomorrow or do some other things. So thank you so much, Tony, as we go on to take the poster presentation. So the model for the poster presentation is simple. We have less than 5 minutes for each, and I'm going to put them up here like a poster. Then if the author is here, he's going to quickly introduce it within 2 minutes. And if there's question, we take in 3 minutes. If he's not here, then it's a poster. So you go through the poster and we move on to the next. So I will start sharing with that of Shewin Elijah. I think he's with Daniel Adewola Adenera. So if you're here, Daniel, you could quickly comment on this If you have need for me to shift to the second slide, I will do for you. If you don't have, then, but everybody, please, you are expected to, maybe I will have to make this a bit, yeah. Is Elijah here? Please, host, you can unmute Daniel Adewale Adediran. Daniel, are you here? It's important you introduce your poster, but we can't wait forever for you. Could you unmute yourself at Ediran? You only, maybe host, please, could you unmute at Ediran, Daniel? Okay. Go ahead if you want, yeah. We could hear you faintly. You can add more volume. You have 2 minutes more.
Research intern at Ellis Bowden Institute and a co-researcher of this research project. I'll be presenting on the bioinformatic approach to the design of multi-epitope peptides in the diagnosis of schistosomal hematobial infection. Schistosomiasis is one of the neglected tropical diseases that has been affecting Africa majorly, and it is especially common in Sub-Saharan Africa and Middle East. There are basically 3 types of schistosoma species that have been affecting human. Then we have the schistosoma hematobium, we have schistosoma japonicum, and schistosoma hematobium. Of these 3, schistosoma hematobium was found to be very prevalent. Presently, the diagnosis of schistosoma hematobium is done by collection of urine samples for consecutive 3 days. With the sign of schistosomiasis at the first examination were compared with the results at subsequent visits. This means that the previous results might not show that this infection is actually schistosomiasis infections. So our research goal is to design a diagnostic kit that will be sensitive and will be specific for these particular infections. These are the methodology that we use in carrying out this research. We receive some protein sequence from the database, then we subject them to some bioinformatic analysis, such as antigenicity test. We did membrane topology test. We did every cell epitope prediction test, and this bioinformatics analysis that we did shows that our construct is okay.
1 minute more.
So these are some of the results that we got from this analysis. This is the construct, the physiochemical properties. The physiochemical properties show that the stability index is okay and stable, and the aliphatic index shows that it is quietly soluble. To the next slide. This is the secondary structure prediction, showing that it is highly antigenic. This is the tertiary structure. Actually, we get like 5 models from this tertiary structure. So this is the one with a highest confidence level. My conclusion, up to this present moment, most of the current diagnostic approaches for schistosoma hematobium are not sensitive and specific enough, and they are quite costly. So from the bioinformatic analysis and evaluation that we carry out, this shows that our diagnostic kit is highly specific and accurate, and it can be available everywhere. I would like to appreciate the staff and management of Ellis Bowden Institute for the privilege and immense support for this research program. These are some of the selected references that I used. Thank you so much.
Thank you, Daniel, for the first presentation. So we move over to the next one.
Thank you for having me.
Yeah, thank you. So the next in our is yeah, I think there were two from Helix. Yeah, from Elishion Olufemi. Are you there? So your time have started counting. 4.5 minutes left. Then you can indicate by raising your hand so that they will unmute you.
Good afternoon.
Thank you. You have 4 minutes left.
Good afternoon, everyone. I am Nnamdi Azikiwe, and this research work is based on designing a multi-epitope peptide for the diagnosis of lower infection using bioinformatic approach. Anyway, lower has been a very, very deadly disease, and it's a neglected yet tropical disease. It's a neglected yet tropical diseases. So these diseases is majorly caused by an organism known as loa. Now, the organism itself has been very, very difficult to diagnose based on its overlap with other organisms, with other infections like onchocerciasis, due to that they were all tested using microfilary. Now, for the testing of the diagnosis, it has always been used of microscopy always. But nevertheless, this work is aimed at getting a more secure, a more sensitive diagnostic kit therapy. What we do is we try to retrieve sequences of viral agency, about 44 of them. And what we have is we take them a series of bioinformatic approaches. Number one, we test for the antigenicity, we test for the antigenicity, we test for the topologic transmembrane, the signal peptide, and a core others. After doing this, what we do is we link them together, then we take them through some post-translational methods. Now, and the post-translational design was subjected to others to check the viability of the constructs we've made. Can I have the next slide? Here is the result. The result shows, the table above here shows the physical properties of the construct we predicted. We have the average length, we have the theoretical p-high of about 8.14. Then we have the instability index. This is a very, very, very interesting stuff because it shows that this is going to be quite stable, and then the construct is going to withstand a wide range of temperatures. So here in Nigeria, that electricity has been a major problem. So what we do is that it can easily withstand temperature, and it can be taken far and wide the country so that the infection of Ebola can be easily diagnosed. And verily, the constructs we try to predict.
A minute more.
The constructs we try to predict is a point of care contact. So I would like to conclude by saying, yet the
Thank you,
epidemiology of Loa is increasing, but the diagnosis of it is not showing for. So with this patent, Loa is going to make it effective. I acknowledge the staffs and management of Helix Biogenesis for providing me with technical support. Here are my references.
Okay. Thank you, Sheun, for the wonderful presentation. And maybe probably those who have questions could write you, as already we have indicated in the abstract. I will move to the next abstract we have, which is like the last one for today. I don't know if Emmanuel Orano is there. Yeah, this is one-page poster. This is very good. Okay, I don't know if you guys are seeing this. Or we go to what it is initially. Is Emmanuel there, Orano? Emmanuel is not here. So yes, so let's go through the poster and have a look, and thereafter I will. So Emmanuel said he's here, but already his time is up. I only wanted to ask you a question. Maybe you just take this question, Emmanuel. Having discovered that butophenol, riboflavin, and the mucuna purines have better binding affinity as a result of your molecular dynamic studies, could you inform if anything has been done in vitro to validate this score, this study, and this wonderful outcome?
No, we've not done any work in vitro yet to validate that.
You don't have any future plans on this?
We don't have future plans to validate it.
What are your limitations?
Funding.
Funding, okay. Okay, thank you, Emmanuel. Nice work for those who went through. I just went through the M and the methodology and the conclusion. Thank you for being part of this. Then for the last today, we have Cheta. There are many others, but I think those that sent their slide, I have to first stop this. Then I share Cheta. Okay, so this is from Cheta. So what we'll do, I will give a minute per slide. So take note of his title. Then Cheta, if you're here, maybe I could allow you. Are you here, Cheta? She's not here. Okay, so let's go through on our own. Next slide. Okay, next slide. And the final slide. I don't know if Cheta is here. Okay, yeah. Cheta is not here, probably to attend to question, maybe one. So I would like to stop this, and I wouldn't know if there is, I think, yeah. So these are the ones that we have. We have already overshot by 16 minutes, and I'm doing all my best to close this Saturday. Cheta is not here. Okay. So please, those who presented the abstracts and the poster, please do send us your profile picture, and we shall get close to you, just as we did promise concerning our grants for your presentation and to encourage what you're doing. Aminu, please host, could you unmute Aminu? I think Aminu have, sorry. Aminu, did you send your slide to us? Host, could you unmute Aminu Abudu Lai? Aminu, I think you are muted. Did you send a slide? Huh? We can't hear you. Host, could you unmute this guy, Aminu Abdullahi? Azubike, could you unmute Aminu Abdullahi?
Aminu is already unmuted.
Aminu, could you talk? You're already unmuted. Okay, thank you so much, Aminu. Guys, you can locate Aminu's abstract in the book of events and read it there. He's having local challenge with the voice. And unfortunately, I did not have his 2 slides. We requested that they should send across. But all those who, we have your abstract projected here. Please kindly send us your profile, then write us so that we can be able to communicate your grants across to you as promised initially. I think at this point, I will want to call on Azubike to unmute himself and also show his face, not this gentle one that is here, and bring a closing remark to this meeting. Then after that, we take the national anthem and have a closing prayer. Maybe I will ask Chenemi to close us then in prayers. Azubike, over to you.
Okay. Good afternoon, everybody. I want to thank everyone that is that made out their time to grace this symposium. I want to thank especially our resource persons and those of them that presented the papers, and of course those that submitted and presented posters. You are the ones that made this event a great one. Also, I want to thank every participant to this symposium. Without your presence, what we are doing here, or what we did here, wouldn't have been a great success. I want to thank the LOC, from the chairman to the list person. And I also want to thank our senior friends, the Vice Chancellor, Nnamdi Azikiwe University, and the chairman of the board, Professor Bioma Nwogu, for their support to what we are doing here. I'm sure that going forward, we'll have much more elaborate outing in the nearest future. So keep pace with us. If you have not joined the WhatsApp group, the link will be posted again. Join us so that you get information about what we are doing, and also keep pace with our social media, and may God bless you. Thank you very much.
Okay, thank you, Anedu. We take the national anthem and we'll close.